Hello, my name is Professor Mike Hughes. Today, in this video, my team and I are going to show you just how simple it is to do electrophysiology using the Deptech 3D.
When starting your 3DEP software, the first thing you'll want to do is turn the kit on, and that's found on a power switch on the back. When the kit's on, the light on the front of the kit will turn on. You'll also hear the kit turn on, and once that happens, you can start your 3DEP software. Once the software boots up, the first thing you're going to want to do is go into Tools, Preferences, and select the COM port that your kit is connected to. The next thing you'll want to do is change your default path to wherever it is you want to save. It's important to save to a folder that you know isn't being accessed by anyone else. So for instance, if you're saving to a shared folder, you're going to want to save to a different path first, because if that's being accessed while the software is trying to save to it, it will cause the software some problems. So once you find your folder, and we'll just save to the desktop in this case, you can then start adjusting your settings. So one of the settings that you'll adjust is light intensity. You'll only need to vary light intensity between 29 and 31 normally. Normally 30 is an okay default setting, but you may find if your sample is highly concentrated or has a low concentration, you may need to adjust this. And we'll look at that in a little bit. Your capture interval is defaulted at 1 second, and you'll leave that alone. These boundary value for automatic fitting, they're tick boxes for fixed values and then unfixed values. These can be adjusted at any time, and you can also do this after you collect the data during your analysis. So you can leave it as its default setting for now and adjust that later. If you want to adjust it right now, you'll just click on the variables that you want fixed, so much like cell radius. And the ones that you don't want fixed, you'll just input a range of values that you'll want the model to fit those to. The next thing, especially if this is your first time running a cell type, you'll want to save images. So what this does is every image is saved every second for this capture interval. This way you'll be able to look back on your data and look at the images to make sure that the kit is working properly. So we'll click Save Images. It defaults to waybands by area, just leave that the same, and you'll click Save. Now you're back at the normal screen for the 3DEP software, and we'll start the experiment. In order to view inside the kit, you'll press Preview Video. Okay, so there's a chip in the kit right now, so if we open that up, you'll see what the kit sees. So if I move the chip back and forth, you can see that happening on the preview window screen. In terms of the light intensity, if we take the chip out, you should get a semi-gray background if the light intensity is okay. Like I was saying before, you can adjust the light intensity, so through Tools, Preferences, and if we lower this to 29, and we press Save, if we stop the preview and then start the preview again, you'll notice now the preview window is bright white. You don't want bright white, so if you see this, you'll need to readjust your light intensity values. So again, Preferences, and we'll set that back to 30. Raising it to 31 will make it darker. Press Save. Stop preview. Start the preview again. Okay? When you're setting up your experiment, you'll also need to think about the frequencies that you're applying. So there's the lowest frequency field and the highest frequency field. If this is your first time with the cell type, what you'll want to do is start from 1 kilohertz and go up to 20 megahertz. So in this field, you'll put in 20,000 kilohertz for 20 megahertz. We typically run our experiments for about 30 seconds. When you would want to make this shorter is if your experiments are time sensitive. So for instance, if you're doing a drug trial and you want as many experiments done as possible in the shortest amount of time, you can make this shorter, provided that your cell types are larger cell types. So what we found is 10 seconds is typically okay for most mammalian cells when you're looking at the data analysis. However, for smaller cell types like red blood cells, and even smaller, we've done some nanoalgae particles, you need to look at longer intervals of time, 20 seconds, 30 seconds. So it's always better to collect more data than you think you need, which is why we tend to take 30 seconds for all of our data types. The signal voltage is 10, which is default, and your chip type normally defaults to your 805 chip. Once you run this frequency sweep, you can then determine how your distribution between your negative force points and your positive force points look. And you can adjust your lowest frequency and highest frequency based on that. So if you're looking at a crossover frequency, or what we call the dispersion, if that happens where you're not seeing any negative points, for example, you may need to adjust your lowest frequency. If you're seeing too many high frequencies, you can adjust your high frequency to be lower. But this is a standard range that works for most cells. Then we'll load our chip and we'll start the experiment. Before running your experiments, you're going to need to make DEP medium. There's two types of DEP medium typically used. This depends on your cell type. The sugars that you use are dependent on what sugars your cell type will metabolize. So typically with yeast, we use demannitol. With most mammalian cell types, we'll use a mixture of sucrose and dextrose. The amounts are dependent on what osmolarity you need. So typically for mammalian cells, we'll be working around 280 to 300 milliosmoles. So the amounts for that recipe are 8.5% sucrose and 0.3% dextrose or glucose. These will give you an osmolarity somewhere in the range of 280 to 300. This is weight percent volume, so your total volume, if you're doing 100 mils, will be a little bit less of your DI medium, and then you'll add your sugars in for a total volume of 100. So if you weigh the sucrose out to 8.5 grams, once you get about 8.5 grams, you can add that to your DI water. Then you'll measure out your glucose. The glucose you add as 0.3 grams, so that's 0.3% of a 100 ml solution. You'll mix all of this into the DI water. You can use a stirrer, you can manually stir it, or if you've got a nice bottle, you can just shake it. And this should all dilute fairly quickly. Once the solution's dilute, you're going to want to measure the conductivity. And we'll need to adjust the conductivity to a starting point of about 10 millisiemens. Again, this is dependent on your cell type. A good starting point, if you've never done a cell type before, is 10 millisiemens per meter. Most conductivity meters measure in microsiemens per centimeter, so this will be 100 microsiemens per centimeter on the meters. This mixture will measure fairly low, somewhere between 5 to 10 microsiemens per centimeter. And what you'll use to adjust the conductivity, typically we use PBS. Now this is PBS without calcium or magnesium. And you'll add little bits at a time of PBS to adjust the conductivity. Depending on your total volume, so in this case we're using 100 milliliters, will depend on how much PBS you add at a time. So in this case we're adding 75 microliters at a time. And you'll just mix it up and remeasure. So even with this small amount, we went from 6 to 27 microsiemens. while this is a slow process, it's better to do it in small steps of the PBS than to do it too high steps and end up going past the conductivity that you need. Okay. And you'll see after 4 additions of these 75 microliters, we're at about 93 microsiemens per centimeter. So I'll add just a bit more. And now we're at 99.4. So that's close enough to 10. The amount that you add will vary from day to day. It depends on whatever the room temperature is. It depends on how accurately you measured your sugars. So don't be concerned if you don't get the exact same number every time or you're not adding the exact same amount of PBS every time. Just go for the conductivity that you need. At this point, if you need sterile media, what you'll need to do is if you're mixing your media in a nice glass bottle like this, you can autoclave the mixture and then you'll want to leave it to sit so that it reaches room temperature. Or you can sterile filter. When preparing your cells for your DEP experiments, the first thing you're going to want to do is get your cells into suspension. In order to do this, if you've got an adherent cell line, you'll use whatever protocol you normally use to detach your cells to get them in suspension. For a suspension cell type, all you'll do is simply pipette your flask into a tube. Once you've got your cells in suspension, you'll need to centrifuge these down to produce a pellet. In order to do this, again, you'll always use whatever protocol you normally use to passage your cells. In our case, with Jurkat cells, K562 cells, and our HeLa cells, we use 1,000 RPM for 5 minutes, and that's sufficient enough to get us a pellet at the bottom of the tube in order to resuspend in our DEP medium. This will make up your first wash. Once the cells are done centrifuging for this first wash, you will then take your cells and remove all of your media supernatant. This is the same protocol that you'll use when you're passaging your cells. It's important to get as much of the media out as possible. You'll want to do this slowly to make sure that you don't disturb the pellet. Once you removed all the media supernatant, you'll have a pellet at the bottom of your tube, and you'll resuspend this pellet in 5 milliliters of DEP medium. You'll want to make sure that you pipette your pellet so that the suspension is uniform throughout the 5 mils of DEP medium. At this point, if you don't know how many cells you've put into your flask, this is the point where you'll do a cell count. Typically with DEP experiments, you'll want somewhere in the millions of cells per mil range. Somewhere between 1 and 3 million cells per mil usually is enough. This also depends on your cell type. If you've got smaller cells, you'll need a higher concentration, with larger cells needing a lower concentration. You can take 10 microliters of this cell suspension and do a cell count while you're doing your second wash. Your second wash will again be the same centrifuge settings as what you use for your passaging of your cell type. Once your second wash is done, you'll take your cells and again remove the supernatant, being careful to remove as much supernatant as possible while not disrupting your cell pellet. At this point, you'll resuspend in DEP medium, but the volume will depend on your cell count. So again, you're going to want somewhere in the millions of cells per mil range. So if your cell count gave you 10 million cells per mil, you'll want to resuspend this in 10 to 20 mils of media. However, most cell counts are going to be lower than that, and you'll end up resuspending somewhere between 1 mil and 5 mil, depending on what type of cells you're using. At this point, your cells are sufficiently washed and all the high conductivity medium should be removed. You can then safely run your DEP experiments. However, if you're still finding difficulty getting results, you can always do another centrifuge wash to ensure that you've removed all the high conductive media. After you've prepared your cell sample, you'll need to prepare your chips for loading the sample into the kit. The first thing I like to do is take fresh DEP medium and wash it into the chip. You'll get a nice little bubble forming on top of the chip, and then you take a lint-free towel and just wipe out all of that DEP medium. This cleans the chip coming out of the box and gives it a nice surface for the cells to enter into the chip. Now, for loading the chip, there's a couple different ways that you can load the chip, and this is completely up to personal preference and your cell type. So for instance, I use red blood cells. Red blood cells are small, and they're used to shear stresses. So using something like a bent needle and syringe works perfectly for the red blood cells. However, if you have a larger cell type, you'll need a larger gauge needle, or if you're worried about the shear stresses on those cells, you'll want to go to a pipette. For the needle and syringe, a 1 mil syringe is sufficient, and I use a 25 gauge needle in this case, and you can see that it's bent slightly. So this is just used taking a pair of scissors and bending. We also cut the serrated part of the needle so that it's a blunt needle. This way, you prevent damaging the back surface of your chip. So if you're using the needle, you'll simply take some of your cell sample and load your chip. Now again, this is completely up to personal preference. Some people like to leave the chip lying flat on the desk, in which case you'll come over top of the chip and in any well that you wish, pipette in your cell sample. And what you'll notice is you get this nice meniscus forming right on top. Now with the flat end of your needle, you can then wipe this across the chip. You'll then take an 18 by 18 millimeter cover slip. So this is a special size cover slip, so you'll need to order these ahead of time. And you'll place that over your meniscus. Now the nice thing with using the needle is if you see any bubbles in your chip, you can then simply push the cover slip out of the way and pop the bubbles with your needle tip. So that's one of the advantages to using a needle. If you're using the syringe, there are advantages to the syringe use as well. For example, if you're very limited on your cell sample, so if you've only got a mil or even less of your cell sample, you'll want to make sure that you keep track of how much volume you're using. With these 1 mil syringes, you can't get a good measure of that, whereas with these, with the pipettes, you can measure out exactly the minimum requirement volume to fill these chips. Depending on your cell type, that's somewhere between 75 microliters and 85 microliters. So you'll take your cell sample, and in this case I'll demonstrate what you would do if you want to hold the chip. So previously I let the chips lay flat on the table, and in this case you can also keep the chip at an angle and you can pipette the liquid in that way. And again, you can pick any well that you want. And then you'll just take the pipette tip and again smooth out that meniscus. You'll take your cover slip and gently layer that cover slip on top of the meniscus so that all of the sample evenly distributes. Once your chip is loaded, you'll load it into the depth reader. In order to do that, you'll angle your chip slightly up, place it into the 3-dep, and slide it just over the pins in the back of the holder. You'll notice that the preview window is now showing your chip. In this case, we have a couple air bubbles. What you can do is pull your chip out and pop the air bubbles with a needle. And then you can place your chip back into the kit. You'll notice on your preview window, it's a nice gray color. So in this case, the light intensity is okay. Should it be really dark, so for instance, if you couldn't see the wells, the outline of the wells, you'll want to change your light intensity, so lower the number of your light intensity to brighten the light. If it's too bright, so for example, if it's any brighter than this, or the background is more white, like the background of your screen, you'll want to higher the light intensity number to make it darker. Once you've got your chips sufficiently loaded, you'll notice that your wells are in the middle of this preview window. You can then lock your chip. And the little lever will press down, inserting the pins into the pin connections of the chip. Before you close the door, you'll want to give this a slight nudge, being gentle not to disturb any of the connecting pieces, but just a little nudge to push everything on top of the pins in case the lever arm didn't go fully down. You'll close the door, making sure that it's nicely sealed, and you'll update your file name and start your experiment. If the door isn't closed properly, you'll get a window to say that it's not closed properly. So what you'll want to do is just push that door closed a bit tighter, click OK, and then you'll need to click Start Experiment again. At the bottom of the screen, once everything turns on, you'll hear a click and it will say Experiment Running. At this point, this status bar will tell you how far along the experiment is. So in this case, we're only running it for 30 seconds. You'll also notice that the kit door, the 3-dep reader door, is now having an orange light instead of a green light. So this tells you that the signal generators are on. Once the 30 seconds has expired, you'll hear the function generator turn off, at which point it's safe to open the door. And you can pull your chip out and reload another chip for the next run. Once the 3-dep reader has finished its analysis, you'll notice that the Analyze tab pops up with the spectra that it produces. This automatically gets saved as an Excel file, an XML file, and an image file of the image of your graph. So you don't have to do anything with this data right away. You can wait until you're done with all of your analysis and then come back and analyze your data later. So in most cases, this screen will pop up and then you'll just press the Record tab and run your next data point. Once you're at the Analyze stage, you'll load your data, your previously saved data, and then this is where you'll get to fit your spectra. So this is where you can start getting the physiological parameters out of the data that you've collected. In this case, you can set your parameters to fixed in your Tools Preferences section. And in the defaults section, and in most cases, you'll fix things like cell radius. So these are things that you can measure either before or after your experiments on a hemocytometer using any sort of image analysis, or if you've got a cell counter that does radius measurements for you. You can take those radius measurements and fix it into your model. Other properties that you can set as fixed are your medium conductivity. So this is why those washing steps are so important. Because we make the DEP medium to a set conductivity, such as 10 millisiemens per meter, once you wash all of the supernatant off, all the high conductive media, you'll know that your medium conductivity is that 10 millisiemens per meter. That can get fixed, and also relative medium permittivity can be fixed, and we use the permittivity of water, which is 78. All of your other parameters you can keep as unfixed, and then this is just where you put in your range. So the default range is meant to encompass any physiological values. However, if you know a range that your value should fall within, you can update these ranges. Once you click Save, you can go back to your Analysis tab and then look at individual data sets. So in this case, we're going to look at bands 6 to 9. And like I said before, you normally only need about 10 seconds of analysis. If you're interested in fitting the single shell model, which is your standard DEP model, you'll click on Auto Fit Single Shell Model, and then you'll click Plot. At this point, you'll notice a yellow line come up. So this is our model. You'll notice the R value is 0.9915, so that R value is actually really good. But you can also start to look at your data points and realize that this data point here is most likely an outlier when you look at your model fit. To remove that, you simply click on that point, and it will ask you, do you want to remove this data point? If you click yes, it removes that data point and then updates your R value. So now we've went from 0.9915 to 0.993. So the fit has gotten better removing that data point. You'll also notice that things that got adjusted were cytoplasmic conductivity, cytoplasmic permittivity, specific membrane conductance, specific membrane capacitance. Your medium conductivity and medium permittivity stay consistent, stay fixed, and then the cell radius is whatever you want that to be. Now, if you've got cell radius measurements that vary, so for instance, you've got a large standard deviation, one thing you might want to consider doing is fitting this model with a range of radius values. So in this case, it's 5, but then if we come in here and tell it 4, you can replot that data, and it replots a new model. So it hasn't changed much, so you can kind of verify that that change in radius measurement hasn't done anything to your model. If it does do things to your model, then you may want to consider saving all of those results. Once you're happy with your fit, you'll simply click Save Result, and everything gets automatically saved to whatever that original save folder that you've put into your default path at the beginning of your data analysis. You get an XML file, and you get a graph, so a JPEG file of your data. So the methods for using the 3-dep reader are relatively simple. However, they do need practice. So I highly recommend practicing on a cell type or a cell line that you have readily available that's easy to culture, easy to handle, and that you've got a high concentration of. This way, you can start with high volumes, high concentration, and get to know how to load the chip, run the experiments, and analyze your data. At that point, once you know how to load the chip and analyze the data, it's really limitless in terms of the cells that you can analyze in this 3-dep reader. In our lab, we've obtained electrical physiological data from things ranging from normal human cancer cell lines, clinical samples, stem cells, small cell types such as red blood cells, animal red blood cells, and nanoalgae.
I hope we've managed to show you just how simple it is to take high-quality, label-free electrophysiology data using the Deptech 3-dep. Around here we have a phrase, electrophysiology is everywhere, but you need the right tool to look for it. The Deptech 3-dep is that tool. If you want any more information or to arrange a demonstration, please get in touch with our distributors at www.labtech.com.